mean, to put things in context, I will tell you a little bit more about the connection between ARAM and our institution. I went to a FASET conference in Maine, and I was sitting by myself on the grass, and ARAM came to talk to me. And we spoke about the 27th degradation and how it would be great to collaborate, putting together our respective expertise. So between 1999 and 2004, we co-authored 10 papers. This is 5 of them. And then in 2004, ARAM was awarded the Nobel together with his ex-students, Aram Chakanover and Gilwin Roth.
I'm very glad to be again back here at NYU, where I spent many summers and one sabbatical in the lab of Michele Pagano. It was very enjoyable and also made some good progress, I think. And thank you very much for organizing this symposium that celebrates the ubiquitin system. I'll try to tell you how it all started and where do I think we are going now. So it all started when I came as a young postdoctoral fellow to the laboratory of Gordon Tompkins in San Francisco to do my postdoc. And at that time, protein synthesis and its regulation were in the mainstream, in the focus of biomedical research. And Gordon Tompkins also worked on the similar problem of how the synthesis of some specific enzymes, liver enzymes, is induced by corticosteroid polymerase. So I thought that this was very interesting and I chose to do my postdoc in this lab. But when I arrived, I saw that it was a large lab, 20 to 25 different postdocs and graduate students worked on the same problem, on the same problem of the synthesis of one certain liver enzyme, tyrosine aminotransferase. And I thought that it is too crowded, and I asked Gordon for a different project, and he said, why don't you work on the opposite process of the degradation of the enzyme? It might be interesting for regulating the levels of the enzyme. So that is how I started to work on protein degradation, on which I'm working ever since. That experiment was done by Aram Chakanover, who was then my graduate student and later shared the Nobel Prize with me for this experiment. And it was in a not very, published in a not very prestigious journal, UBRC. Usually you don't get tenure for such a publication, but I think it was an important paper because it was in the first step in the right direction. So we used lysase from reticulocytes, in which, as you see, the degradation of a protein, for example, radiolabeled globin, was strongly stimulated by ATP. So we got the reconstitution of energy-dependent protein degradation in the test tube. And now we began to fractionate it. We fractionated it, just put two very crude fractions. Fraction 1, which is all the proteins that are not bound to the param, and fraction 2, which are all the proteins that binds to the resin ion exchange resin and diluted together with histone. You can see in fraction 1 there was no degradation in the absence or presence of ATP, and in fraction 2 there was some degradation, but most of the ATP stimulated degradation was lost. But we found that we just added back fraction 1 and fraction 2, there was a complete regaining of ATP stimulated protein degradation. So that very simple experiment told us that the system that degrades proteins independence of ATP is made up of at least two components. Some components are in fraction 1, some in fraction 2, and you need to add back both of them in order to get the complete activity. So that was the first step to the right direction.
So we've heard a lot about reconciliation from Aram, who's the discoverer and founder of this field. But my background is in clinical genetics. I'm interested in monogenic diseases. And something that really has struck me about ubiquitination is the fact that many different genetic diseases are caused by defects in the ubiquitination pathways.
I think our research can fit in. A couple of years ago, together with a group of young scientists here at NYU, we identified the ubiquitin ligase, SPOC, as the physiological ligase for this MyD88 protein. And in that publication, we showed that SPOC can target wild-type MyD for degradation, and that's important for resolution of emergency hematopoiesis. And our work was also then followed up by a number of groups later on that confirmed our finding.
What we found out is that LZTR1, it is not a transcription factor, and in fact, it is a protein that contains these KELPS repeats and TRuB back domains. And I don't need to educate you much on this, but as you probably know, when proteins have this domain architecture, they are generally adapters to the Queen 3 ligase through the BTB back domains. They can bind to POL3, and then the KELPS they tend to form these couplers. So after a while, a few experiments, we actually demonstrated that LZTR1 is the adapter for the POL3 for grade 1 for proteasome degradation.
It's also a different kind of network protein that it doesn't interact with the columns, at least as far as we can demonstrate thus far. So no interaction with col1 or any of the other columns that are going to interact with FOX proteins, but instead it interacts with this protein called NPP2, which is a large protein, and hopefully we'll get a chance to talk about that at some other point. FOX045 lives at 3Q29. At the terminal end of 3Q, way at the end, there's a microdeletion syndrome, which others have shown, which encompasses FOX045. Whether it is implicated in disease or not is not quite known. It's about the true mega-base deletion that occurs heterozygously in these patients.
So as Aaron nicely introduced already, Protease is targeting chimera, PROTAC. It's a bifunctional small molecule that induces target protein degradation.
And then this term obviously now become relatively popular in the drug discovery world. We know that there's an increasing number of clinically relevant drugs represented by IMiD compounds, and also in this one, a sulfonamide compounds, and then a number of other compounds emerged in the literature. And different from the bifunctional PROTAC compounds that's presented by Jen earlier today, microglues are monovalent compounds, and they're much smaller, drug-like, and they don't have to show very high affinity to at least one of the protein partners in the system. That opened up this opportunity for us to sort of target a protein that's unligadable.
Now, you're familiar with this modular architecture of ubiquitin ligases very much, and basically, from our point of view, this is very attractive because all the substrate are recognized by these discrete substrate recognition domains. So we can imagine that, for example, DHL recognizing its own target can be repurposed to target something else. And this was originally developed by Kota's lab in Dundee, where they fused anti-GFP nanobody and showed that they can degrade GFP target proteins.
And my lab, in fact, is focused on understanding the biology of a substrate receptor of the CRF3 family, known as KIP1. Some of you can be familiar with the substrate receptor because with this well-established substrate NRF2 are the master regulator of the oxidative stress homeostasis. And the reason why we became interested in this substrate receptor is because it's encoded by a tumor suppressor gene often mutated in non-small cell lung cancer. And over the year, genomic study has demonstrated that about 20% of non-small cell lung cancer patients carry inactivating mutation in HIP1.
And this is a protein called UB2, and we got interested in UB2 because it is very important for ribosome homeostasis, which is something that BioLab is very interested in. And so it has two functions related to ribosome homeostasis. The first one is, of course, ribosomes are made of about 80 different proteins that all have to assemble together. In mammals, we don't have operons, so these proteins are all individually made. And so what UB2 does sometimes is that if there are extra copies of a ribosomal protein, one function of UB2 is to essentially target some of these for proteasomal degradation by ubiquitinating them.
So we started working on this technology. We call it TransTAC, or transferrin receptor targeting chimeras. So this is a bispecific antibody technology. So basically, these are very molecular design. We generally use a Ig1 kind of molecule, and then with the blue domains are the anti-protein interest binder, and they can be antibodies and antibodies. We heard about monobodies earlier today. It can even be ligand, peptide ligand, or active domains that come from natural proteins. And on the C-terminus of these proteins, we have the anti-TFR1 antibodies. So at the cell surface, we think TransTACs will bridge a target protein to TFR1 induces rapid proteomization, and protein engineering approaches such as cleavable linker, or I'm going to talk about other mechanisms later as well, with this going to decouple the two. So TFR1 recycle, a target protein will be able to get to degrade through the lysosomal pathway.
Wonderful symposium. There was an excellent mix of basic science, which I still believe leads the way, and application, and you will use it. And it's now Louis, 21 years from the Nobel Prize, but it's 45 years for the discovery, 25 years before, and we learned a lot. We still have to learn a lot, but I think the time has come, the time is right for it, using all the knowledge that we learn to put it into human health and to health discovery. So good luck to all of you, and thank you.